welcome, and thank you for attending today's webinar with Fullscript and Dr. Chris Miletus on identifying food sensitivities across culturally diverse diets. My name's Kaylee, I'm with Fullscript, and we are very excited to have Dr. Miletus here with us today. Before we get started, I was going to do a little housekeeping. We want to let everyone know that this is being recorded, so you'll be sent a recording to your email as soon as after the webinar is finished. You can go back and watch it again. It's also live, will be live on our website, fullscript.com/webinars. You can find all our past webinars there as well. And throughout the webinar, if you have questions, please add them to the question section on your control panel or your dashboard there. We will get to them at the end of the presentation for a little live Q&A. And so with that being said, I'm going to disappear for a little bit and pass it over to Dr. Miletus so we can get everything started.
Well, thank you, and thanks for the great introduction. Thank you all for joining, and it's great to see such a nice international group of like-minded providers. So many of you already know, my name is Dr. Chris Miletus, naturopathic physician. I've been very blessed to write 16 books, a couple hundred national articles. I proudly say I was institutionalized for 7 years as the Dean and Chief Medical Officer at the Naturopathic University here in Oregon. I was blessed to have my colleagues vote me a physician of the year in 2003, both for our educational outreach at the Naturopathic University, starting the first residency program that was accredited, but also for starting 16 clinics for the poor, which I continue to do in my clinical practice to this day. So with that said, we all are contributors to the healthcare system, and we all teach one another. And it's with that humility that we'll have this conversation. The information that we're going to share will, of course, be just free-flowing stream of consciousness as one clinician to another clinician. You will see on the GoToWebinar platform, you can type in your questions. It should look kind of like that. And at the end, we're going to read the questions and answer them. Also, you'll see at the last slide, I have my email address I've had ever since 1996. And if you have a personal question or if you want some of the information that I'm sharing as a handout or other things, I can do that as well. So today's topic, selecting the appropriate panel, will of course help better determine what is fueling our body and our patient's body and what's hindering. And I've had patients when I've done food sensitivity panels, and I've had them on the supplement of turmeric. And lo and behold, they have a sensitivity to turmeric. Or as we've seen the rage, all the rage of CBD, well, what happens if they have a hemp sensitivity and it is a hemp-derived CBD? Is this something that we want to take into consideration? And of course, we're all guided by the concept of do no harm. So once again, it kind of lets us guide our patients accordingly. And the more we try to spice up our life and eat internationally and realize that each culture has so many health benefits and traditions have kept them going for millennia, it's like, wow, but is that the right one for me? So we're going to compare and contrast various health panels or food sensitivity panels. The Asian panel, as it's called, the Japanese panel, the Hispanically influenced Mexican panel, the vegetarian panel, and then some a la carte generic panels as well. Of course, there's all kinds of panels out there that we can measure for food sensitivity. And also, as we've moved towards a Zoom medicine with COVID-19, and I hope it doesn't become COVID-20 or COVID-2021, and we can all kind of pray and mindfully kind of ponder, may this virus go away. But because we are doing a lot more Zoom medicine, I have been using a lot more finger prick tests where I have it drop shipped to my patient and they collect the blood spots for the 96 or the 144, the 208, or the Asian or Japanese or Hispanically influenced or vegetarian panels, which I'll have a visit right after they collect their sample, we get the results, and it allows the practice to go seamlessly as much as we can these days. So we'll also discuss inadvertently sabotaging wellness protocols by recommending a botanical or recommending a food which is good or is a fad. But once again, as Lucretius said, the Roman philosopher, what is good food for one person is poison for another. Of course, we all know Hippocrates, and of course, give even time between the Romans and the Greeks, and of course so many other well-versed Eastern medicine philosophers and physicians and providers as well. But Hippocrates says, may your food be your medicine be thy food. But the Roman Lucretius said, but it might be poison for you, even though it's good for everybody else. So, and that's kind of the concept here. So voila, here's me. I'm the product of two immigrants. And indeed, I benefit from my mom coming from Germany, adopted from an orphanage after World War II in Germany, where she spent in Munich the first 5 years of her life with the bombs going off and so forth during World War II. And then my father's from Greece, and they met as exchange students. And kind of how that works here in Oregon and all other states and parts of the world is voila, here I came. And so now the question is, am I more guided genetically by my German heritage or my Greek heritage? And most of us are a blend of many different cultures and many different strengths, but also genetic susceptibilities as well. So teasing that out, and because we are such a beautiful melting pot in the westernized world, but throughout the world, we have to kind of ponder, the next time I go out for Vietnamese food or Chinese food or Thai food or Greek food or German food or Ethiopian food, you name it, am I eating within my genetics, personal susceptibility, not just for my ethnicity, but also my individual epigenetic manifestation. So I have my genetic predispositions, but then I have my epigenetics. Oh, how many antibiotics have I taken? Do I have fluoridated and chlorinated water? Have I been eating organic all the way along? And of course, our microbiome determines quite a bit of that as well. So it's not always the usual suspects. Of course, we all think, oh, well, it could be dairy, it could be shellfish, it could be the peanut. It could, of course, be gluten. But I think many of us have had patients cross the pond over into Europe, and I've had patients which have had to avoid gluten, and then they go over to Europe and they can't pass a little Italian or French bakery or bistro, and they're like, oh, I was so naughty over there, Dr. But I felt great. So is there a differentiation there? And some people argue it's glyphosate. Some people argue it's a different kind of wheat. But once again, knowing that we are a product of what we eat from our head to our feet. That was dating myself from the 1970s cartoons, you're what you eat from your head to your feet. Circa the same time, I'm just a bill on Capitol Hill wanting to become a law. So that's when actually we learned something from cartoons. So what's interesting from an educational perspective is about learning, and of course we all know the Latin word discere, doctor is teacher. So under resources at US Biotech, you can actually see a whole lot of webinars on demand. But at the same time, you can look at food panels, and there's a wide variety of food panels. You can have a 96 food panel, an Asian panel, Japanese panel, the Mexican Hispanic influence, so also South America, of course, and 96 vegetarian panel, which was really important for a family member of mine, which I'll share a case study. And then of course, and those can all be done through a simple finger prick test, as can the 208 and the 144. Now, if you're doing IgE panels, those are serum draws by the nature of the fragility and lability of IgE as an immunoglobulin. It just isn't very stable, so it needs to be done and processed in a certain way. But lots of these can be great utilities for sustaining and growing our practice. But also what's interesting, a presentation I've given was about how IgG, which is once again how we trigger complement. And our complement pathway, part of our immunological pathway, is of course critical for fighting off viruses. So I give the example, do I tell my patients, do I want you to be fending off against an artichoke or an asparagus that you've all of a sudden fallen in love with because you heard it's healthy? Or do I want you to be having your IgG ever vigilant and non-distracted for a virus that might be coming around? And so once again, when we do IgG testing, I've been doing a lot of it in my practice this year because I've been doing zinc testing, of course, vitamin D testing, other, of course, nutrient testing, but I've also been doing the food sensitivity so that patients know that they are eating towards wellness and not distracting their immune system. So the first patient that we're going to chat about, and interesting, how did I determine which panel to run for her? 51-year-old female, presents to the clinic with burping, flatulence, body aches, lots of tendinitis, lots of autoimmune-like symptomology. And lo and behold, we found out that she's positive ANA, and we quantified it to 1 in 2,500. So as you know, we go 1 to 1280. So she was 2560 on her ANA. So she was cranked up there. And now the question is, she's feeding that self-attack. And we also found out that she also had Hashimoto's as well. She reported eating the standard American diet, but one of the benefits of the time we spend with our patients, we dig a little deeper than, okay, well, just what do you eat? And we discovered she had Hashimoto's. She also was diabetic with an A1C of 12.8. And lo and behold, why was that? She identified as a vegetarian, but 70% of her food was carbs. So lots of times like, well, I'm a vegetarian. Are you? Or you're a carbivore instead? And so lo and behold, we have, of course, had to work on that. But then the question is, because she does not eat meat and she does not eat overt eggs, though she'll have it in a baked item, what do we do to support her? And also knowing that she had a limited budget. So on a normal panel that we might consider in our clinical practice, you say, okay, great, we're going to do dairy, we're going to do grains, we're going to do vegetables. But lo and behold, we wouldn't necessarily want to do fish for her. That'd be wasted antigens. And likewise, she's not a meat eater, and she does do a little bit of egg. But so if I can replace those on the plates that are being ran with something that's broader and more specific for her, wouldn't that make great sense? And so that's what we did, was we went ahead and did this vegetarian panel for her, which offered the dairy, because she happens to also be a big fan of dairy. And in fact, her big argument was, I really don't necessarily want to know what my food sensitivities are, because you're not going to get me to eat meat. And if you take away my dairy and you take away the items with egg in them, I'm not going to want to eat. And she enjoys planning her day around where does she get to go out to eat. So here we got the dairy, but look, now we got more vegetables, we got more grains, including some alternative grains, which is important, some fruits, and she's, as much as she's a carbivore, she's also a fruitarian, if there's such a word, because once it's summer season here in Oregon, oh boy, does she go for it. And so we kind of want to look at offering the most specificity as possible. So she enjoys asparagus, she enjoys eggplant, she enjoys millet. And then also with the Hashimoto's, I want to know, well, are there anything else that I can offer her in terms of proteins and or an alternative grain, so like quinoa? But we want to know, well, great, I'm going to just say, we're going to go gluten-free, we're going to do more rice. But is it rice the right choice, or is quinoa the right choice? So we're going to try and go gluten-free. And lo and behold, in her case, she acknowledged over the course of time, because I have some really nice cross-reactivity sheets, love to share with you. And so at the end, my email will be present. But she, ever since she was about 13 years old and went through puberty, she couldn't eat melon. Prior to that, they would eat watermelon, cantaloupe, and even cutting up a melon for a family picnic causes her hands to itch. But lo and behold, cantaloupe and melons cross-react with ragweed that cross-reacts with echinacea. And whenever this patient was ill, they would often take one of the many immune products out there that had echinacea in it. And lo and behold, echinacea cross-reacts with ragweed. So I have a cross-reactivity sheet, more than glad to share with you. And my contact information will be at the end. Likewise, what about dates? What about grapes? What about jackfruit? Kiwi. And of course, kiwi is part of the latex family, so cross-reactivities. Mango, persimmons. And so you get the idea, lots of different foods that are on a vegetarian panel that would not necessarily be on a general panel. So if you know a person's not really a big meat eater, you might choose this way, but there's some other options which we'll talk about. And there's a few more herbs on a vegetarian panel than there are on a 96 regular panel. But then we go to my 40-year-old executive, and she is a challenge. She's a different patient. She went from 225 pounds down to 159 with intention, and now she's gone back up to 240, 250. But she lost weight. She did great. But something went on. She had surgery. She had a breast reduction. She had some infections. Her microbiome got disturbed, and she got a job promotion. She succeeded in the corporate world. But the only problem with the corporate world is she leaves at the crack of dawn. She has 2 kids at home, which are very active in sports and competitive dance. So lots of stress. She has a history of migraines. She has very intermittent periods, and she's also has a partner that introduced her to Panamanian food because that's part of his ethnicity. So she's kind of eating a hodgepodge. So I had to figure out, could I be feeding her microbiome improperly, or could she be feeding her microbiome improperly, or could be triggering inflammatory processes which are altering the ecology within her GI tract? And so we went for looking at the Mexican or Hispanically influenced foods to see whether or not we could find a culprit for her. And so I often challenge my patients with the concept of, so when you eat, who do you feed? And of course, my patients go, duh, I'm feeding myself. I'm saying, no, think of your little microbiome, the 100 trillion little bacteria in your GI tract and other microbes. Think of them as fledgling little birds and your mama bird feeding them. They often eat before your body does and the cells do throughout your body because they're living right there at the source of the food. And so, of course, we know Firmicutes and Bacteroides, and that ratio can affect obesity. And that's one of the things which we were targeting for her, is to change her diet, change her inflammatory markers, and avoid things which were a culprit. And voila, it's helped her remarkably. And so once again, that's called the Mexican panel, and it has a little bit more Hispanically influenced concept behind it. But then we go to a 30-year-old marketing guru, and her partner is from Korea, actually, and they eat a lot of soy, which of course I had a lengthy conversation about the downsides of soy and GMO and all of that. But then she also eats zucchini beans and mung beans. And so the question is, do we pick a Japanese panel, an Asian panel? But overall, she's doing well, but she is looking for lowering inflammation, getting some GI harmony going on. She would get bloat, and many of us know of a condition called Rome-Held syndrome. Rome-Held syndrome is when the vagal nerve gets stimulated because of distension within our GI tract. It could be in our esophagus, it could be our stomach, it could be SIBO, or it can be lower intestine, whether it be the 25 feet of small intestine or 5 feet of colon. But we know that 80% of that vagal nerve is afferent or sensory. And hence the vagal nerve is called the vagabond nerve because it's going down throughout the whole GI tract, but it goes right along our esophagus and of course to our heart. So we can start getting heart palpitations, dizziness. And I wrote a paper on that, which you can ask for at the end. Once again, my email will be there. That speaks about Rome-Held syndrome, the vagal nerve, chronic fatigue, fibromyalgia. So we were trying to figure out, is the food she's eating, can we get rid of the bloat so she wouldn't be dizzy, she wouldn't be faint, and she'd have better concentration? And so we had to look at which panel. So on the left-hand side, you see the Asian panel, as it's called, or on the right-hand side, the Japanese panel. So in the last maybe month, because here in Oregon, on the county that I'm in, we're allowed to go out to eat up to 8 p.m. and then all the restaurants close here in Oregon. And so you have to be masked up and take all the precautions and be 6 feet apart. And now there's plexiglass in all the restaurants. But once again, we eat around the world on a regular basis. We've eaten Vietnamese, we've eaten Japanese, we've eaten Thai, we've eaten at a Mediterranean restaurant, a Lebanese restaurant, and of course we do other fare as well. So I'm having to do a very broad panel, which we'll discuss later for our family, and we have. In fact, I've measured my 27-year-old son at the age of 7, at the age of 18, and at the age of 25, because food sensitivities do change as our microbiome does, as our GI integrity does, and our stress levels. Because when we know we're stressed, and who is in today's world not stressed? Of course, here in North America and the US, we had elections. We have, of course, border issues on both sides. We have COVID. Many parents have become home teachers, or basically teaching their kids on top of their normal functions, and we don't have that same social interaction. So whether you're a homeschooler or not, you know there's a lot of stress in the world. So that's affecting secretory IgA, because we know when we get stressed, our IgA levels lower, and this can make us more susceptible to not only viruses, but also alter microbiome. So here's the Asian panel, here's the Japanese panel. I'm not going to go through it in any great length, other than there's differences. And for example, over here on the right-hand side, there is duck, lamb, and over here on the Asian, there's also goose. And if someone's really into their goose, then of course you want to make sure it's cooked right, pun intended. And likewise, on the Asian panel and Japanese panel, there's nuances of whether or not you have certain foods that are going to eat or not eat relative to seafood. And as you can see, the Asian panel is a little broader, surprisingly, than the Japanese panel. And also the herbs are a little bit different as well. So of course, we're trying to help our patients in the obstacles in the course of life, but it's hard to avoid what we can't see or we're not aware of. And so in the case of one of my family members, every time, and it's a young family member, every time they ate salads, they felt terrible. And so when we went ahead and fed them salad and said, well, go ahead and eat your salad, it's good for you, they said, oh, it makes me sick. And I think many of us have childhood traumas, for lack of a better word, around certain foods that we were forced to eat that just either palate-wise or flavor-wise just wasn't our thing, but it was eat it and clean up your plate. And of course, I'm 55 years old, so that's my generation. It wasn't a discussion. And so I said, oh, no, you got to eat your salad. It's really good for you. It's making me sick. Okay, well, how could crunchy water make you sick? So vinegar, a vinaigrette, ranch, just eating it by itself with some salt on it. Lo and behold, first time ever in my clinical practice, I had a patient quantified right justified on IgG to lettuce, or what I term crunchy water. But we didn't know that was one of the things altering his microbiome and altering his vagal nerve and contributing to dizziness and not feeling very well. So once again, we need to quantify that, or what I say, test, don't guess. So there's a 96 food general panel. There's cultural diverse panels, and we are a melting pot. We are a combination genetically, each one of us. And with that genetic uniqueness comes our strength and diversity, but also comes a degree of susceptibility to certain foods that either weren't over the millennia within our gene pool's eating habits, or a unique combination of the blend of whom we are. So there's 144, 208 panels. But one of the things I point out to my patients, and it's been a really big growing opportunity for me in my practice, is I've actually been able to grow my practice during this period of time, and we're more successful than we were even prior with using of Zoom and doing drop shipping, which a laboratory will do for free to your patient. They collect the sample and we get the results. So what we've created wellness protocols, and we get them tested, and we make sure that we're not triggering complement. So if you see here, the C1Q, the beginning of the complement pathway, IgG1, IgG2, and IgG3 all activate complement, but so do viruses. So do I want to be worried about artichoke, or in the case of a family member, lettuce? Or do we give them a break on lettuce? It wasn't good for them anyway. It's triggering that immunological response, and we minimize that. And the answer is clearly yes. And some other interesting tidbits here is look at the half-life of IgG1, IgG2, IgG3, which are the combination of the IgGs and the food sensitivities offered by this laboratory. Is we see 60% prevalence, 32% prevalence, 4%, and we got the IgG4 there, which we'll talk briefly about on another slide. But look at the half-lives. There's a memory to those, that lettuce reaction or that asparagus reaction, or maybe it's egg or dairy, or the more common ones. So what we want to do is see, well, how broad can we cast the net? So this happens to be a 208 panel. So it captures a lot of the ethnic panels and a lot more. So for example, let's say we're trying to be super healthy. And so we're eating a protein bar that's sweetened with dates, but maybe there's a date reaction, or maybe there is a reaction to another item like blackberries. Well, that's low on a glycemic load, glycemic index, and we're working with a person with metabolic syndrome, but blackberries aren't good for them. Maybe they had a big reaction, not in this case, as you can see on the sample report, but maybe it's blackberries aren't the thing for them. Or likewise, maybe mangoes aren't, or as you can see the stars coming here, tangerine. And so the benefit of a larger panel, which I like to do in my clinical practice, and once again, it comes down to the budget. We can only test as much as the patient's able to afford and flex towards their wellness. But when we think about food, I try to get them to go up to their comfort level to identify what might be going on. But you can see here on seafood, and of course I live in Oregon, so I'm along the Pacific coast, so there's lots of seafood. So do we eat bass? Do we eat catfish? And of course, if you're down from the south, you know that you might go noodling for catfish. And some would argue, if you're not familiar with that term, you go thinking about catfish. Now, noodling is actually where you stick your arm into the muddy bank of the river and have the catfish grab onto your hand or your arm. So that's a whole nother level of entertainment for those that have time. Sea urchin, not on my list of things to eat, could be quite the prickly experience, but if you're so inclined, you'd like to know, is sea urchin good for me? Is it sustaining me? Is it adding to my health savings account, as I tell my patients, or is deleterious? And on the dairy, this is what I really like about the 208 panel, or broader panels in general, is the fact that if I take away cow's milk, can I allow them to have sheep cheese, sheep milk, goat milk, and if only they would add donkey milk, because donkey milk, there's some research showing it might be the most close to human milk, even closer than goat and sheep milk. So go figure that one, and who did the research on donkey milk. But I remember giving a lecture on that about a decade ago. But identifying, if I take a food away that might be a source of calcium or protein, can I bring something back? So that's the benefit of differentiating the dairy in my mind's eye. Chia seeds, beyond the chia pet, which you might have gotten as a gift somewhere along the way, chia seeds are very good, very faddish right now, and I would argue rightfully so. But once again, what happens if a person had a problem, or kola nut, or macadamia nut, or pistachio? And one of the other things I have is a handout that you're welcome to ask for that actually gives a list of histamine-rich foods. And when we think of food sensitivity, so often our patients will say, but I know I react to that food. But do you react to that food IgA, which of course think mucous membranes, as you can see in the white lines here, IgG via the complement pathway and some other pathways, or do you measure via IgE? And I differentiate for my patients. A food sensitivity is something other than IgE, and a food allergy in the allopathic world is, of course, a food allergy because that's IgE driven. I say E is, think of it as E for emergency room, and think of G for gradual. So you can eat a food that you're, of course, sensitive to, and we all know this, and I eat it today, but then all of a sudden I feel crummy on Sunday. And that's very common for an IgG food, or also what we'll term as the stacking effect. But histamine, so lots of nuts, seeds, smoked foods, other foods are high in histamine. I have a nice little chart, be more than glad to send to you. My email will be at the end. And once again, quinoa. Once again, if I'm going to take away something, do I add something back? And if I'm doing a replacement, am I blindly replacing a known high food with a food that may or may not be safe because we haven't tested it? So beets, of course, we know beets are all the rage with nitric oxide. We know by the age of 50 for gentlemen, about 55 for women, our nitric oxide levels are less than 50% of where they were in our youth. So our vasodilatory effects aren't there. Our immunologic or regulatory effects aren't where they used to be. So beet powder all over the place, but should the patient be doing beet powder or eating beets to support nitric oxide? Once again, quantifying that. Kelp, of course, we know iodine levels have dropped for the NHANES studies in the United States by 50% from the 1970s to the late 1980s. And we even failed the World Health Organization when it comes to people of reproductive age and iodine status, and therefore thyroid status, and also potentially altering IQ, a lecture that I give on iodine. So should I be doing kelp? Should I be going to have sushi? Should I be, once again, kale and kelp? And likewise, okra. As we know, there's many reasons a patient may have symptoms. It might be the IgG, IgA, IgE, might be histamines. It might just be that these foods, like pistachio, is high in histamine. Or maybe it's high in lectins, L-E-C-T-I-N. And so lectins, of course, Dr. Gundry and others are speaking a lot about lectins. Dr. Nutritionist Carolyn Perini out of California, a decade before Dr. Gundry made lectins a common word for a lot of our patients, was speaking about lectins. And the lectins are another way our patients get irritated, either from beans or nuts or seeds or wheat germ agglutinin, another reason for non-celiac driven wheat issues. But okra is a common ingredient in a lot of the supplements used to bind up lectins. But okra in and of itself can be problematic. So once again, identifying, well, why am I not getting better? You said it was possibly a lectin, but indeed the treatment might be part of the problem unless we know. And also what you'll see here on the right-hand side, I'm going to jump over here for a moment, is a Candida screen, or some like to say Canada screen. And it is not diagnostic, but it lets us know, am I reacting to Candida yeast, or Candida, as you might want to enunciate it, as part of the problem? And if so, is that a cross-reactivity? Because I'm doing a lot of kombucha fermented foods. Is it cross-reactive with Saccharomyces boulardii? Maybe that's not the best supplement for me. Or indeed, do I have an immunological reaction because either myself or my intimate partner has some yeast and I'm getting oral contamination or auto-inoculating myself? And so this can lead to, okay, well, let me do some stool testing. And you ask some questions about oral contact with potential yeast sources. So once again, in this case, they have both an IgA and an IgG score here, because the practitioner had ordered the IgA, IgG, and IgG4. So once again, looking at whether or not we can replace duck egg, can I have bison or buffalo if someone was so inclined, lamb. So once again, think the Mediterranean ethnic foods. I'm skipping rabbit for a reason because I had a pet rabbit, so we're not going to talk about eating rabbit. And so, or venison. So maybe they're out hunting and gathering literally from the land. And same thing with the grains. How about brown rice? Lots of my patients say, well, I have, okay, on white rice is okay to do brown rice. Or can I do millet? Or can I do sorghum? And of course, we know sorghum is a big controversy because it's put into so many things. And or can I do teff? So as we take away, is it safe to give? And likewise, black pea, black-eyed peas, mung beans, flaxseed. I think it's all the rage. Flaxseed oil, flaxseed, one of my favorites. Whenever I do a higher carb food, I will actually have ground flax seeds put onto my food, 2 or 3 tablespoons. I'll sprinkle them on, because it actually is making it a high fiber meal and it lowers my glycemic impact. Let's say if I was to have a hash brown breakfast with some hash browns, potatoes, high glycemic index, but you can lower the load by putting ground flax seeds. But if that was a problem for me, that would be a bad thing. Likewise, we all love certain herbs like ginseng, or using hemp, or using hops. I think also beer here. And of course, hops and cannabis, hemp, sativa, or marijuana are related to hops, or shiitake mushrooms. Maybe we're using them therapeutically for immune support, but once again, it could be problematic. And once again, we like to spice up our food. I'm not going to spend a lot more time on this, but you can see all the different spices, or maybe licorice tea, or oregano, or maybe some sage, or likewise turmeric, all the rage, curcumin. So once again, we want to look for those things. And of course, the candida, which we were talking about. So when we have IgG foods, my approach when there's a score of 0 to 4 is I try to allow my patients to eat the zeros freely, and then the 1 scores once every 4 days. And that's my optimal. Some people have too many scores, and then I have to moderate that. Or if a person's on a biologic drug, so like a drug for rheumatoid arthritis or psoriasis, or maybe they're just got a corticosteroid injection into their shoulder or their neck or their back, or they're using an oral steroid or inhaled steroid, then we're going to realize that if I do the test and they can't get off their steroidal nasal spray or whatnot, we're going to have some dampened scores, and then I'm going to have to clinically adjust them up if they're underreacting. But if they're on a biologic drug or an immunologically suppressive med, we also want to go ahead and look at, are they still reacting even while on that? And then it's dead to rights that these foods are problematic. So on the IgG scores, I allow my patients a 1 once every 4 days, 2, 3s, and 4s. I try to have them avoid it. I'm going to work on some gut wellness. And they say, but do I really have to avoid it? Can I do both at the same time? My clinical approach is I want to avoid irritating the GI system while I'm trying to work on leaky gut syndrome, permeability, reestablishing the microbiome and ecology. And so I'll use glutamine and collagen and probiotics. And I will use inositol-glucosamine. However, because inositol-glucosamine is usually shellfish derived, crustacea derived, I will avoid it or be very careful where I source it from. And so if they have a crustacea issue, a shrimp, shellfish, etc., I will have to go for a vegetarian inositol-glucosamine. And likewise, some of our patients will end up presenting with a shellfish allergy. Like when I did my food sensitivity test more than once now over the years, because I've been practicing for 28 years, I've done it for myself. And invariably, I either have a crab or a lobster reaction. Well, that's important because one of my cross-reactivity sheets, I'm more than glad to share with you, shows that dust mites cross-react with the Crustacea family. So it's like, okay, well, I have a tremendous dust mite allergy. I got allergy shots as a kid. And so I was growing up in a Western model. And so it's like, okay, go get your allergy shots, go get your allergy meds. And so it's like, okay, there's a cross-reactivity there. But also think about chondroitin sulfate for joints or glucosamine sulfate, all crustacea derived, unless you have a very specific vegan or vegetarian source. We could be undoing some of that. And likewise, I'm a huge fan of digestive enzymes to cut up what I call for my patients the barcode. So think of a barcode on any of the items that you might be buying at a store. Now, lots of us can self-check out. So you scan the little barcode. Well, that barcode is like the amino acid sequence of an antigen. If you can cleave up that barcode or put a line through it or clip up the barcode, it's not going to be as readable to the immune system. Once again, it doesn't give a pass, as I tell my patients. It's something to consider. I never want to see an IgA reaction that's meaningful on a food sensitivity panel, because think our eyes, nose, throat, lungs, GI tract, or female reproductive regions all clear with IgA. Stressed out, it's lower, and that's why we do, once again, an IgA total serum if we're looking for, like, say, celiac disease, to see is there enough reactivity. But if I have a person with higher than a 0.5 score on IgA, I try to have them avoid it, because once again, I don't want to have those mucous membranes irritated from the mouth all the way through the exit, because once again, it shouldn't be reactive, and so we try to avoid that. And then we get to probably the most controversial of all the immunoglobulins for food reactivity, and it's the IgG4. In our allopathic model, you would think IgG4 is considered a blocking antibody for IgE, and to a lesser degree for IgG. So, oh, but I have a high IgG4, and therefore it's okay for me, quote unquote, to eat XYZ food. I clinically disagree, because if I'm having to spend a lot of energy producing IgG4 to deal with a food, it's really something I want to deal with. But I can send you a great article about eosinophilic esophagitis and IgG4, and as well as some other research, but definitely for our eosinophilic esophagitis patients, our IBS and IBD patients, there's clinical research showing that IgG4 can actually be the protagonist, can actually be causing the problem, and it's not necessarily protective in those cases. It could be causative, or at least correlating with the symptomology. And the same thing that's been found in some subcategories of autism is that IgG4 may be making things worse, because it's stimulating the immune system at a fundamental different level. Simplicity-wise, and I think we all know this, and I'm speaking to colleagues here, is there's fixed reactions, there's earned reactions, there's cross reactions. So fixed reactions, you can thank mom and dad for it, just not going to go anyplace. It's always going to be your weak spot on a given food. And so whether that be a true allergy, IgE, or it's an IgG or IgA, it's just your susceptibility or the proverbial Achilles heel for your body. And think about, why is Johnny or Jill born with a peanut sensitivity or a true anaphylactic allergy or an egg allergy, or why do they develop it early on in their life, and why is that pervasive in a family? So that's why I term fixed. Earned is when we've overconsumed them. We just eat them day in and day out, and you'll see those scores are inflated. And lots of our patients will challenge us, well, this only picked up the foods I eat. Then I tease them out on that conversation. So you look at a food panel and you say, well, do you eat a lot of garlic? Well, I eat garlic. Do you eat garlic as much as you eat dairy? Because they're similar scores. Oh no, I do dairy every day. So your garlic score is equally as high as your dairy score, but you don't eat it every day. So what does that tell you? And then he says, do you eat onions? Oh yeah, I eat onions all the time. So why isn't that one high? So we tease that up, because at first blush, it will seem like the foods we eat are often the higher ones. Part of that's just because in a westernized diet or an ethnic diverse diet, we have a tendency to go to our favorite foods. And it's said that the average person eats the same 25 foods day in and day out. And I challenge my patients on that statistic. I said, well, tell me, what foods have you eaten? And I'm going to be silly here. Well, I had French fries, of course, not under my watch, but I had French fries, I had tater tots, I had potato au gratin, I had mashed potatoes, I had baked potatoes, I had hamburger, I had steak, I had, so I would say, so you had meat and potatoes. Well, but no, they're different. Not So we fall into our trends. We do eggs different way, we do chicken different way. You get the idea. But it's kind of fun to gently nudge our patients to think about that we all fall into ruts and eating a broader diet, more phytonutrients, all the things we know. But then we get to cross reactions. So for example, if you have a latex reaction, maybe you put a Band-Aid on and you don't respond well to having adhesive on your body, or a more dramatic latex, well, bananas, avocados, other things cross react. Or up here in Oregon, we have, of course, lots of birch trees, and birch trees have white bark, and there's certain foods you should not eat if you're out and there's a lot of high birch pollen. So if you go to pollen.com, Pollen.com, type in your zip code, you can actually see what pollens are going high or low, and there's several other indices out there that you can also search for. But if there's a known reactive inhalant out there, it can make the foods that a person's dealing with worse. So lots of times our patients are doing better, they're doing better. I'm losing ground. I don't know what's going on. Maybe it's spring, maybe it's fall, and maybe it's mold and mildew, maybe it's the birch trees. Or as in the case of a family member, it was ragweed, and that made the melon and the echinacea that much more worse. So once again, looking at that cross reactivity, or the proverbial straw on the camel's back, is absolutely essential. So in my practice, and we all have our preferences, but I identify whether or not a person has some leaky gut hyperreactivity and I try to deal with that. If they have a lot of food sensitivities, I'm working on gut microbiome and helping to heal the GI tract. And this is just a simple approach to glutamine, collagen, probiotics, and acetylglucosamine, digestive enzymes. Then the question is, when, if ever, do I retest? So I'll avoid the reactive foods, usually wait closer to 6 months. I'm doing gut repair at the same time, and then I'm going to remeasure what's going on. And lots of times, if there's a lot of what I call seesaw pattern, highs and lows throughout all their foods, I'm going to do also a GI test of some sort to make sure there's not a dysbiosis, which is leading to leaky gut. And we know that butyric acid and butyrate, short chain fatty acids made from our bacteria in our GI tract, are absolutely essential for the mitochondria within our enterocytes and colonocytes to heal, to do their prime function and help with those tight junctions. So I'm making sure the butyrate levels are dialed in, and then I'll remeasure to see how we resolved the foods. And then the question is, do I have my patients go ahead and eat all their bad foods again and see, do a provocation test, or do I measure them on all quiets on the Western Front? Avoid the foods we knew and we've identified, and then remeasure them and see which scores have gone lower, knowing that they're always going to probably be a weakness for you, but have you been able to quiet down your immunological system? Most of my patients, I'll just measure them on all quiets on the Western Front. I don't reintroduce the foods, but if a person's desperately wanting to reintroduce a food, like I've got to get back to my eggs, I got to go back to my cottage cheese in the morning, then I'll have them do a little provocation to see what's going on. And this is a nice little kind of example I share with my patients. And if you want these slides, you can also email me. I'm more than glad to share them with you. But I tell my patients, guess what? I got a picture of you. Oh, okay, well, maybe it doesn't look exactly like you, but on Tuesday you ate a food. And then on Wednesday you ate the food again. And let's give it, on Tuesday you had an omelet. On Wednesday you had quiche. On Thursday you had heavily dipped non-gluten French toast. Well, you've created a reaction on Tuesday. It's going to last 72 hours plus. Then you recreated the reaction the next day and the next day. And so as a result, you have a stacking effect. And for those people that are familiar with the ocean or large bodies of water, I say this is like waves. There's two ways to get your feet wet: walking along the shoreline. One large wave coming and crashing along the shoreline, so it'd be like a very high food sensitivity, or lots of little waves stacking up just right. So imagine if you had a low score to pistachio, but you do pistachio Tuesday, pistachio Wednesday, pistachio Thursday, it's going to have that stacking effect, and voila, you've ended up getting ready to sacrifice yourself to that food in my stair-step example. So if you want these slides for educational purposes, I'm more than glad to share them, because this is what I incorporate into my Zoom business with my patients, is I explain all this, then I put in the results, and it's like a whole little presentation I customize for them by just kind of throwing in their results. There's a lot of on-demand presentations I've presented, Dr. Andreas presented, available. Literally, you just go to usbiotech.com, you click on whatever one you want. Lots of references, lots of literature supporting things like demystifying cross-reactivities, or mental health in the wrong food, or about phthalates and parabens, or there's one that I originally titled Don't Die Dieting, but it's talking about testing for environmental pollutants during weight loss. We will be liberating fat-soluble toxins in our fat, and that can tip the scale on top of our daily exposures to, of course, ill health. And of course, leaky gut and catch-22 of GI disease, right-hand bottom corner. Dr. Andrea, phenomenal colleague of ours, actually has presented extensively on COVID testing. And so if you want to know more about COVID testing, she is the go-to. She works with US Biotech. And so as you can see here, multiple ways to look at COVID and why does COVID hit different organs. She goes through all of this and the nuances of the pathophysiology. And I give a lecture that I gave back in March at the beginning of the COVID-19 event here in the United States about how creating homeostasis within our mitochondria, because there's a substance called MAVS, M-A-V-S, and as a mitochondrial receptor activity, and viruses seem to stimulate this and actually determines how many cytokines were created. So your mitochondrial health is important. So I do a fair amount of organic acid testing as well to make sure the mitochondria are dialed in. We know that for every decade from the age of 30, we lose about 10% of our ATP production. So I'm 55, so I'm 25% behind the curve, all things being equal. But we also know biological versus chronological age. Likewise, NAD, critical, where so many enzymatic pathways of not only the citric acid and carbohydrate metabolism, but also immunological support. NAD levels drop dramatically when we actually get a virus that's attacking our body. And NAD levels can drop up to 50% from the age of 40 to 60. So another little talk that I give. But the beauty of all of us working together as colleagues and the fact that we're a melting pot of society is we have the ability to eat from the wisdom of all the cultures, but we have to make sure those foods are good for our body. And it might be good for mom and dad and one sibling, but not all the siblings. So we've all seen that in clinical practice. So I'm going to open it up for questions and turn it over. And thank you all for your willingness to come and listen to me talk on this topic. And I'm available for further questions along the way. And as you can see here, my email is naturalmedman@hotmail.com. That's the one I've had since 1996. I'm attached to it, even though as I've grown older, it's a little bit more corny every year that goes on.
I think it's great. All right, we had quite a few questions come through. It's really engaging. I think I am definitely a carbivore, if I have to say so myself. But we got a little bit of time, so let's see. And a few of the questions I think were asked early on, so you may have covered it, but I thought maybe it's good to repeat quickly here. One of the first ones we got was, I was curious about how IgG antibodies are just a normal immunologic response to foods and not necessarily a sign of a food intolerance or sensitivity. What I continually hear is that sensitivity tests may be misleading and make people remove foods they don't necessarily need to take away. Can you explain this further?
Certainly. And so it's a very great question. It's an insightful question. And it's also, let's not all be sheep question, which I think is really important in functional medicine and medicine in general is there any research on it? And in fact, I have a lot of research, more than glad to share with anybody on the peer-reviewed literature on how IgG food reactivities link to depression, migraines, and a myriad of other things. I will say that some people will have IgG scores and they're quote unquote asymptomatic. They have no overt symptoms yet. Their vitality, or as we say in naturopathic medicine, the vis matrix naturae, it's sufficient that they haven't succumbed or it's not bad enough to hit their threshold. So some people will have IgG reactivities, but the question is, why does our immune system react to some of the foods in our diet and not other foods, is my philosophical question. And once again, I think it's a great question. And I think it's something which we have to be wary of, not to just take foods away from people. And when we do take foods away, because there's an inside wisdom in the question that was asked, is that we take dairy away or protein away, we could be doing harm. And so therefore we have to really look at, if I modify a person's diet, how do I replace and still provide all the macro and micronutrients and not also cause so much stress in one's life. And one of the things I tell my patients is when you do an IgG or IgA test or IgG4, it is giving you information in the buffet of life to make conscious choices. And it doesn't necessarily have to be a millstone around one's neck. Great question.
Yeah, that's great. So this next question is a little longer too. It has maybe a couple of questions in it. But are the foods sourced on an IgG panel organic or conventional? Could the body be reacting to the pesticides or chemicals in the foods? And if our client eats over 90% organic, is it still fully relevant in your opinion?
Yeah, so I'm a big fan of organic. All of our ancestors prior to Industrial Revolution was largely organic, minus any creative ancestral experiments. And so, of course, we're all familiar with the Dirty Dozen foods from the Environmental Working Group that they update every year, ewg.org. And so Dirty Dozen foods clearly have to be organic when the budget allows. Of course, there's the Clean 15 foods, but in terms of organic, these are highly purified antigens going down to the molecular level where, yes, they are vastly organic when organic antigens are available, but they're also highly purified to specific things like casein or dairy. One could make an intellectual argument that if something's not organic, could there be a carry-along molecule that's a contaminant theoretically? But I think that organic, if your patients are doing organic, which is awesome, and it's what I encourage my patients to do, and our family flexes our budget accordingly as we're able. But there still could be a problem with the food reactivity. And as you can see here on this slide, what we're doing with the ELISA technology or also chemiluminescence technologies, we're creating arranged marriages within these little wells of a one-time interaction and seeing what is their level of reactivity. That's oversimplified, of course, many times over, but I would say that it's still germane, but I think that I find that most of my organic oriented patients have less reactivity unless they have a tremendous dysbiosis going on.
Awesome. So next we have, are there any allergy panels that differentiate between or A1 versus A2 dairy? We may have talked about that.
Not that I'm aware of, but once again, I'm just a practitioner out here, but not that I'm aware of. And
Okay.
and if you do know, feel free to email me because then the next webinar I give, I will let people know.
Yeah, perfect. We're all learning here. So this is from one of the attendees. Do I see an elevated IgE levels of a certain food when I eat too much of it? Or would I see?
Ooh, IgE, so true allergies versus a food sensitivity. I would say that if you eat a food and you just had a reaction to it, the answer would be yeah, you probably would see an elevation. It also depends on your total IgE levels in your body. So much like when we measure a person that we're going for celiac disease, we're either going to do an IgA or an IgG, celiac endomyosal antibodies, transglutaminase, whatever it might be. We always want to know what the total levels are, like US Biotech offers both IgG and IgA celiac testing. So it's if your person has really low IgE for whatever reason, so once again, IgE, E is for emergency, kind of E, then I would say that you should expect a higher reaction. But once again, it depends on immunological competence would be my answer. But you would expect it should be climbing at some level because your body should be reacting if you're susceptible to an IgE food reactivity.
Okay, and how do you use adrenaline testing with food sensitivity testing?
I actually don't. But you know, in terms of having an EpiPen available for a bad reaction, yeah, of course we have an EpiPen in our office, but I don't incorporate adrenaline testing as part of my practice, and that might be a weakness of mine. Which, if you have insight as to why that would be good, let me know. Natural Midman at Hotmail, but don't have an answer for that one.
Okay. So this is a little longer one too. I've often heard that IgG results can vary from lab to lab. How do you ensure a lab has accurate and reputable practices? What should we look for to determine it will provide us with accurate and consistent results?
Excellent. Well, you know, in some part, longevity of the company. Of course, any laboratory we're using must be CLIA certified, CLIA certified. But then we look for additional levels of certification like COLA or other accreditations and reproducibility. One of the realities is human error does exist. So the more automated something is, that usually the more reproducible it is. And it's also lab standards as well. When I have visited US Biotech over the years, I've been using US Biotech since 1992. That's how old I am and how long I've been practicing. I have visited every one of their laboratories. And likewise, I've done the same thing for Labrix and other laboratories I use. I will visit facilities because I want to know what's going on behind the scenes. If you were to visit US Biotech, you'll see their whole upstairs looks like a spaceship, and it's largely robotic. Of course, lots of human beings there monitoring it all, but it's about reproducibility. So technology, CLIA, COLA.
Yeah, that definitely makes sense. So kind of along the same lines too, this question is, there are many food sensitivity tests on the market. Are they all created equal if they are evaluating the same IgE, IgG, IgA? If not, how do we weed out the good tests from the bad?
A somewhat politically loaded question. I'm an independent educator for US Biotech. And if you were to come to my office, I have a wide variety of lots of laboratories. I've always chosen US Biotech because of their reproducibility, and they run their tests in duplicate. So what they'll do is when your sample comes in for your patient or yourself, it's barcoded, it's ran blindly through their machinery, their technology, and then unblinded, and the results are compared to see have there been internal reproducibilities. If there's not, then it goes to quality assurance. Many laboratories only measure the test once, a sample once. So US Biotech measures things twice. So whichever laboratory you choose to use, make sure they run things in duplicate, because once again, errors can happen. And when it comes to food, and that earlier question, should we just be willy-nilly altering people's diets based on a test? Let's make sure it's reproducibly so, and that the quantification's there. And actually, there is a 2010 paper I can share with you that compared US Biotech to another laboratory. And the reproducibility, it was blind. US Biotech did not know, the other laboratory did not know that they were being tested. And this was way back in 2010, and US Biotech did exceptionally well. Not paid to market or advertise US Biotech, but these are the reasons I've chosen them for this particular test.
Yeah. All right. Quick last one. Kind of a double one. Does insurance cover testing, and can, say, a holistic nutritionist order labs from US Biotech?
Yes. Myself and Dr. Andrea visit with certified nutritionists and other providers all the time that ran US Biotech tests. It's also available throughout Canada directly to you, so you can order directly through US Biotech. And so the answer is yeah, lots of accessibility there. As to insurance, IgG testing is not generally recognized by the ICD-10 coding as anything that they want to cover. And of course, insurance companies are in a business to save money, even if it's short-sighted on their part. IgE testing, I do believe that there's some remuneration, but I can't speak to that. You need to talk to your account manager. But I think IgE testing does have the ability to have insurance, but I don't know if US Biotech is participatory in that process or not.
All right, there's a few more coming through. So as you said, I know you gave your email. You can also email webinars@fullscript.com, and we can pass along the questions. As I said, everyone will be getting a copy of the recording, and you can find it on our website as well at fullscript.com/webinars. So I just want to say thanks for everyone attending. Very informative, very engaging, and I had a great time. So thank you, Dr. Miletus, as well.
Well, thank you. And as we all go out and enjoy the diversity of the world in which we live and eat broadly, let's do it with good knowledge. That's my thought.
Yes, I agree. So again, thank you very much, and enjoy the rest of your day, everyone.